Good afternoon once again, and welcome to your subject in 2022 Agricultural Biology. So for this video, we're going to talk about the next topic, which is plasma membrane. Last time we discussed about the cell structure, so please take a look and on the video posted here about the cell. And so for this video, we're going to talk about again the plasma membrane. So we all know that the plasma membrane is it's a very important part of the cell, as it is so-called the gateway to the cell. It's where all the substances go through inside the cell and enable for the metabolism and the different chemical activities of the cell or of the plants takes place. So this is a photograph of a cell membrane. It's under a very high-tech microscope. So this is how a cell membrane looks like. And the cell membrane is flexible and allows a unicellular organism to move. So as its flexibility will be discussed as we move through all the different discussions. So, and a very important that we have been mentioning since the beginning is called homeostasis, wherein this is a balanced internal condition of cells. It's also called equilibrium or balance, and it's maintained by plasma membrane, controlling what enters and leaves the cell. So it's the plasma membrane's main function on balance or homeostasis. So the other functions of the plasma membrane as well is as protective barrier. It regulates transport in and out of the cell, which is very selective because it doesn't let all the substance come inside the cell, so that makes it selectively permeable. And this also allows cell recognition and provide anchoring sites for filaments of cytoskeleton. So some more functions of the plasma membrane: it provides a binding site for enzymes. So enzymes also reacts with substances, and we all know from the previous discussion we have that enzymes, they have a binding capability, and they make use of the plasma membrane for binding. So next is interlocking surfaces bind cells together. So it's also where junctions or combination takes place, and contains the cytoplasm or the fluid inside the cell. So again, this was the structure of the cell membrane, wherein it consists of the protein molecule, the carbohydrate chain, the lipids bilayer, and protein channel. And it has the following components. We have phospholipid bilayer again, the cholesterol, the proteins, peripheral and integral proteins, the cytoskeleton, and the glycocalyx. So this, so once again, another photo showing you the plasma membrane. So the phospholipids make up the cell membrane and contains two fatty acids that are non-polar, this one, and head is polar. The head is polar and contains a PO4 or group or phosphate group and a glycerol. So we call it the fluid mosaic model. It's the plasma membrane contains fluid because individual phospholipids and proteins can move side to side within the layer like it's a liquid. and it's mosaic because of the pattern produced by the scattered protein molecules when the membrane is viewed from the above. That's why it's the plasma membrane. Membrane is a fluid mosaic model. The cell membrane have again polar heads, which are hydrophilic or water-loving, while its tails are non-polar or hydrophobic or water-fearing. So it makes the membrane selective in what actually crosses in and out the membrane or the cells. So these are the aqueous environment, and since the polar head are hydrophilic and the non-polar hydrophobic fatty acid tails with the polar hydrophilic head. So only water enters into this side and not into the hydrophobic side fatty acid tails. So again, this is the cell membrane. The cell membrane is made of two layers of phospholipids, this one and second, called the lipid bilayer. And the hydrophobic molecules pass easily in the but in the hydrophilic do not. So in terms of solubility, materials that are soluble in lipids can pass through the cell membrane easily because they are soluble in lipids or phospholipids. So as a semi-permeable membrane, the small molecules and larger hydrophobic molecules move through easily, like oxygen, carbon dioxide, and water, because these are hydrophobic. So as you can see here, the watery extracellular fluid on the hydrophilic and the hydrophobic side, and the another hydrophilic side, or and the water cytosol. So as again a semi-permeable membrane, the ions, hydrophilic molecules larger than water and large molecules such as proteins do not move through the membrane on their own. So they are assisted actually through types of transport across cell membranes. So what are these? We have the so-called simple diffusion. It requires no energy. Molecules molecules move from area of high to low concentration. So this is actually an interactive video, but I'm going to show you a video later to show you the different active and passive transport. So diffusion is passive process, which means no energy is used to make the molecules move, and they have a natural kinetic energy. So they just move freely. That's diffusion. So the diffusion of liquids such as this, dye is dropped in water and diffusion begins, and the dye is evenly distributed with the water molecules. So that's a simple example of the diffusion of liquids. So the diffusion through a membrane actually just solute moves down concentration gradient from high to low concentration. So from high concentration, the solute moves to lower concentration inside the cell membrane. So aside from diffusion, we also have the so-called osmosis. It's the diffusion of water across a membrane from high water potential to low solute to low water potential or high solute. So take note, high water potential have low solute and low water potential have high solute. So diffusion across a membrane through a semi-permeable membrane. So this is what happens in the diffusion of water across a membrane from high water potential or low solute concentration to low water potential or high solute concentration. So we have this so-called aqua proteins. These are water channels, protein pores used during osmosis. So water, these are water molecules, and these are the so-called aquapurines in a model. So cell in isotonic solution, um, the direction of water in the cell is at equilibrium, since the environment have sodium chloride and 90% water and 10% sodium chloride, while inside the cell is also have the same, so 10% sodium chloride and 90% water, so there is no net movement, so equal and equilibrium. While cell in a hypotonic solution, and again this is isotonic solution wherein there's no movement in and outside the cell, but for a hypotonic solution, the direction of water movement is this in going inside the cell, because the solution of here of the solute sodium chloride is lower than here inside the cell. So it has, and it has more solute here in 20%. So the movement is going through inside the cell. So in a cell in hypertonic solution, wherein as you can see, the movement of water is going outside the cell because the solute, or 15%, which 15% is higher than inside the cell, which is 5%. So cells in solutions with osmosis, if the condition is external solution is hypotonic to cytosol, so the movement of water is going inside the cell. But if the cytosol is hypertonic, so the movement of water is outside the cell. And if the cytosol is isotonic, so there is equal, no movement at all. So in an isotonic solution, no net movement of water and equal amounts entering and leaving, while hypotonic solution will result to cytolysis. So since all the water will go inside the cell, and in the hypertonic solution, all of the water will goes out of the cell, so that causes plasmolysis. So the cytolysis and plasmolysis are explained further here, where in cell it's placed in distilled water, cell swell and burst, resulting to cytolysis. While cells placed in a concentrated salt solution, the cells will shrink and shrivel, so resulting to plasmolysis. So osmosis also takes place in red blood cells with isotonic red blood cells, hypotonic red blood cells, and the hypertonic red blood cells. So what happens to blood cells is that in a hypertonic or high solute concentration, so the water, the red blood cell shrinks into water and it's also in the cell. While in the isotonic, no difference, and in the hypotonic, all the water surrounding the cell moves inside the cell and resulting to bursting, for example. So this is again the red blood cell, where hypotonic, hypertonic, and isotonic. So you can again in the this is inside the cell. So this is hypertonic, isotonic, and hypotonic. So the three, you can do this as an exercise, no? Later, label each environment in the spaces provided. So there are three forms of transport across the membranes. We have the passive transport and the active transport. As you can see, this passive transport involves simple diffusion and facilitated diffusion. So in the simple diffusion, there is no energy required. It simply moves from one place to another, or from higher concentration to lower concentration, or in hypotonic to from hypertonic to hypotonic in an hypotonic condition. Well, facilitated diffusion is the passage of materials aided by both concentration agent and by transport proteins. So in an active transport also, molecules again move through that transport protein as with um, using energy or ATP to move against their concentration gradient. So again, in a passive transport, simple diffusion doesn't require energy, moves high to low concentration like the oxygen, water, diffusing into cell, and carbon dioxide, diffusing out. But for a passive transport, it's a facilitated diffusion, which the facilitated diffusion doesn't require also energy and uses transfer proteins to move high to low concentration, such as glucose or amino acids, moving from blood into a cell. So proteins are critical to membrane function. So it takes like a structural support, recognition. So it must be recognized first by the facilitated protein transport, and then communication occur, and then transport is facilitated. So there are also types of transport proteins. We have channel proteins embedded into the cell membranes and have a pore for materials to cross. And we also have another carrier proteins can change shape to move material from one side of the membrane to the other. So these carrier proteins are more flexible than the channel proteins. And in the facilitated diffusion, molecules will randomly move through the pores in channel proteins, such as this. And some carrier proteins do not extend through the membrane, so they band and drag the molecules through the lipid bilayer and release them in the opposite side. So other carrier proteins can also change shape to move materials across the cell membrane. But in the active transport, it requires energy in the form of an inosine thiophosphate and moves materials from low to high, instead of high to low, against a concentration gradient. So examples of an active transport is pumping sodium ions out and potassium ions in against strong concentration gradients called the sodium-potassium pump. So in this sodium-potassium pump, 3 sodium ions pump in for every 2 potassium pump out and creates a membrane potential. So we also have another we call moving the big stuff through exocytosis. So it's moving things out, no? As you can see in the figure, molecules are moved out of the cell by vesicles. We call them transport vesicle inside the cell that fuse with the plasma membrane. And this is how many hormones are secreted and how nerve cells communicate with one another, and as they are released into the extracellular fluid. So this is an actual footage of an exocytosis, exocytic vesicle immediately after fusion with plasma membrane. So it's very nice. Moving, again, another large molecules move materials into the cell by one of 3 forms of endocytosis. The figure 1, then 2, and then 3, and 4. We also have the so-called pinocytosis. It's the common form of endocytosis which takes in the cell molecules as a vesicle. That's pinocytosis. Cells forms an invagination, and materials dissolved in water to be brought into the cell called cell drinking. Another example is the transport across a capillary cell in this blue portion wherein the pinocytic vesicles is formed, and this transport on the membrane. We also have another type, the receptor-mediated endocytosis. So some integral proteins have receptors on their surface to recognize and take in hormones and cholesterol and others and then they are transported inside the vesicle. So this is how the receptor-mediated endocytosis shown on the figure. So we're in this one are being are facilitated by receptors, and they are engulfed inside the plasma membrane. So another endocytosis or phagocytosis is used by bacterium to engulf large particles such as food bacteria, etc. into vesicles. It's called cell eating. And this is also done by lysosome. So phagocytosis, which is about to occur inside the cell. So phagocytosis capturing yeast in this cell, yellow cell, by membrane extensions of an immune system cell in the blue color cell. So exocytosis is the opposite of endocytosis. Large molecules that are manufactured in the cell are released through the cell membrane. So inside the cell are vesicles and the plasma membrane outside the cell. So this is exocytosis, wherein they engulf or take out the molecules outside the cell. And to do this, so this is just the opposite of endocytosis. So I have a video here to show you an active transport animation, which is very helpful for you to remember.
The smallest unit of life is the cell. In order for life to be sustained, nutrients and gases must be able to pass into a cell. Wastes and other products must be able to pass out of the cell. Many of the materials that pass in or out of the cell do so by passive transport. In this process, there is no energy, no ATP expenditure by the cell. Concentration gradients allow for the movement of materials. Osmosis and dialysis, both specific types of passive transport, use this concentration gradient of high to low. Another type of passive transport is facilitated diffusion. Since it is passive, the high to low concentration gradient is present. However, the process is facilitated by a carrier protein. The carrier protein is somewhat like a revolving door, allowing easy access across the membrane and then rotating back to the other side. All of these processes, osmosis, dialysis, and facilitated diffusion, are passive. The cell does not have to use any energy to move materials by these methods. The concentration gradient is used. However, there are materials that need to be moved against their concentration gradient. This process requires energy, ATP, and protein carriers that are found in the plasma membrane. Defined active transport is material moved from a low concentration to a high concentration through a membrane using a protein carrier that requires ATP. An example of active transport is the mechanism of uptake of iodine by the thyroid gland. Iodine is essential for the manufacture of hormones that influence body metabolism. When you eat a meal containing iodine, the iodine will pass from the small intestine into the circulatory system. As the capillaries pass through the thyroid gland, iodine diffuses out of the circulatory system. Iodine attaches to protein carriers in the thyroid cell's plasma membrane. Being proteins, they have a unique shape, so only iodine will fit. Glucose, which is also actively transported into the cell, relies on a different protein receptor. The energy required is somewhat analogous to a rock being pushed uphill. The processes of simple diffusion, osmosis and dialysis, facilitated diffusion, and active transport allow for most all of the small material, those dissolved in plasma and extracellular fluid, to pass in or out of the cell. Large particles, however, pass into the cell by endocytosis. This is an active process. It needs ATP. In this process, the plasma membrane surrounds the particle, such as bacteria or a food morsel, and forms a vesicle that pinches off from the membrane. The cell does this in two different ways. One way is called phagocytosis. In this method of cell eating, large particles are surrounded when the cell extends the plasma membrane and engulfs the particle. The result is a membrane-bound package of material. Fluids that the cell wants to take in enter by a process called pinocytosis. In this type of endocytosis, the cell membrane sinks in, the material sinks inward, and the membrane pinches off. The cell uses energy for this movement. In both types of endocytosis, the cell ends up with a membrane-bound vesicle. The material in the vesicle still has to pass through the membrane in order to be used by the cell. In this case, the lysosome joins with the vesicle and digestive enzymes break down the material to particles small enough to pass through the membrane and into the cytosol. A specialized endocytosis called receptor-mediated endocytosis also occurs in cells. In this process, molecules of a particular substance, such as cholesterol, bind to receptors in the plasma membrane. The vesicle is formed as the membrane sinks in. Then a lysosome joins the vesicle and the digestive enzymes break down the substance, releasing it from the receptors. The substance diffuses into the cytoplasm, the lysosome reforms, and the vesicle, with now empty receptors, becomes part of the plasma membrane again. This allows for the cell to reuse or recycle the receptors. A function of some cells is to manufacture or synthesize materials for export. In many cases, this material is made up of large molecules like proteins that cannot pass through a plasma membrane. The process of exocytosis enables the cell to pass this material out of the cell. The cell is a very dynamic living entity. It is essential that materials move in and out of the cell. This is done by passive transport using a concentration gradient and active transport, endocytosis and exocytosis using ATP.
So that's all for about the plasma membrane. So thank you for watching the video, and if you have questions and comments and clarification, just comment on the posted video, and I will see you again on the next video. Thank you.